Continuing here with our dangerous E. coli 0157H7 strain. We were almost done, but got cut short a little bit. So we were down here on average about 60 deaths per year in the United States. Mostly younger children or older people are the ones who end up dying from this, but anybody could die potentially, but it's just most dangerous for the young and the older people. The infectious dose means how many bacteria would it take to get infected, is estimated to be as low as 4. That's 4 cells, as in 4 E. coli cells, and that's not much. Recent years, the outbreaks were tied to lettuce grown in California, and also there was some outbreaks at Chipotle that almost took that restaurant down, but they recovered, and it's really bad reputation if you end up being tied, if your restaurant is tied to lettuce that was tainted with this particular type of E. coli. So here, moving on to E. coli grown on different media right here. So we have selective media here, EMB agar, eosin methylene blue agar. That causes E. coli to grow in this metallic green kind of growth. You can see it right here outlined. So you have this green metallic kind of growth. That's typical of E. coli. And so this would allow you to differentiate E. coli from other Enterobacteriaceae, because on EMB agar, E. coli grows in this green metallic kind of growth. And then here also, the bottom of an EMB agar, you can see sort of the dark, very dark kind of growth. On McConkey agar, you would see E. coli growing in pink. And here's some bad marketing. If your name is Elmo coli, you might not want to have, I don't know, maybe you should not open a fine food store, or at least don't market like this. I mean, that's just not good advertising. But anyway, just a joke. Our last pathogen for this pathogen group number 7 is Pseudomonas aeruginosa, another bacterium that usually doesn't do anything as long as you are not already immunocompromised or get it at the wrong place. So it's a gram-negative bacillus, gram-negative rod. It is strictly aerobic, so it only likes to grow in the presence of oxygen. It has a polar flagellum, as you can see right here. There's the polar flagellum. It is water-soluble, has this water-soluble green pigment, and you might have noticed sometimes if you have a bad cold and then you develop a cough with it, or you have snot coming out of your nose, that is green, that green snot, that is because of the growth of Pseudomonas aeruginosa. So Pseudomonas is mostly known as an opportunistic pathogen, and so if you have a bad cold and your respiratory epithelium is damaged from the virus, then Pseudomonas takes the opportunity and starts growing there, and that's when you have the green snot or green pus or green sputum coming out. So on agar, and I believe that you mostly saw this actually in the lab, that if we're growing the Pseudomonas, then you have this green growth in your streak plates or in the slants, and that what makes it actually really easy to identify. Some of you had this particular bacterium as your unknown number 1, and it was actually easy to see because if the slant turns green, and you pretty much know what you have. Pseudomonas is among the most drug-resistant of bacteria. It is difficult to treat in general because your immune system has a really hard time finding this bacterium. It has a capsule a lot of times and makes it very difficult for your immune system to see and to find antigens that it could produce antibodies against. Here's somebody with a hot tub dermatitis due to Pseudomonas infection, and here's somebody with a fatal systemic Pseudomonas infection. A lot of people that are immunocompromised, they actually end up with lung infections and with Pseudomonas, and those, they basically will have to learn to live with that infection for the rest of their lives. It's just very, very difficult to get rid of the Pseudomonas once it's in the lungs. Actually, this is here our last pathogen for pathogen group number 7. It's Klebsiella pneumoniae. This one is a gram-negative bacillus with a capsule. You can see the capsule right here. You can see the outline, this halo around the actual cells. That would be the capsule. Let's make this more clear. So this is, of course, a negative stain, and you can see how the bacteria, the cell bodies, they got stained, but the capsule didn't pick up the stain because it's waxy and it doesn't pick up the stain. But here you can see the cells in the center right here, and then you have this halo around the cells. That's the capsule. Of course, Klebsiella pneumonia is the cause of pneumonia. And here on a gram stain of somebody's sputum, you have Klebsiella pneumonia in the sputum of this person. And we can see the gram-negative rods. You have them down here and other places as well. Okay, that's it for pathogen group number 7. I will put this one together with pathogen group number 8, and then we'll have the next lab quiz on pathogen group 7 and 8 combined.